so much for the invitation. It's really a pleasure seeing the friends here, and it's a very great opportunity for me to talk here at BC Cancer Institute, considering the vast experience and the central role that this center has for the Theranostic. So as a disclosure, I'm Senior Clinical Advisor at NIH, National Institute of Health, and also I'm President of Institute of Nuclear Medicine and co-founder of United Theranostic. I'll very quickly start by mentioning that the core of medicine has been the art of caring using best available tools through the history. In this painting by Dr. Field, you see parents that are sitting on the back of this room, and they know their daughter is sick. They know that she may not see the morning. And the doctor in this painting only give them one thing, and that's reassurance that if there is something possible to be done, I'm going to do that for you. That's all that they need at this moment. So that is all that they need at this moment. They need to here. So what about this?
Side now. I'm trying to get the audio better.
Okay, maybe I should just be closer. Okay. Technology is important for both clinical care and lecturing. So my point is that the core of medicine is that art of caring, regardless of which tool is in our hand or not. And that comes with the trust that is between the patient and the physician. Throughout the history, different technologies and different aspects of modernity introduced different things in medicine. Mechanical engineering brought stethoscope. Many people think that's a symbol of medicine from eternity. That's not the case. Before the 18th century, late 18th century, there was not such a thing as a stethoscope. Electrical second revolution brought electricity, and electrocardiogram and related technologies are there. The third industrial revolution, of course, brought the computation and the connectivity. CT and PET and many other technologies are out of that. And right now we are at the verge of the fourth industrial revolution. And at the end, it doesn't matter because this is only a new tool in the armamentarium of the medicine. But there is a new kid in the block, the Theranostic. Theranostic is by far one of the fastest growing aspect in a healthcare industry. It's not very uncommon that we see a merger or acquisition of one of the companies with $1 billion valuation. Fusion acquisition by AstraZeneca, Raise Bio by Bristol, and Point by Eli Lilly is among those matters. And the market expansion is astonishing at this moment when you look at the investments, both at the new drug developments or commercialization of that. But that's not just like the financial aspect of that. Look at the scientific and clinical medicine. The number of articles that each day we see coming to the field is growing semi-exponentially. And number of clinical trials the same way. We have significant number of clinical trials coming in this field. So when we have all of these amazing movement, we should just have a perspective. A few years ago, this diagram from the Oppenheimer came out that showed many of the companies in various phases. As you see, the outer segment are the molecules that are in preclinical, and then when you go from the phase 1, phase 2, phase 3, and the clinically approved or FDA approved. Very recently, we have the update on that. You see the number of companies changed, and some of the molecules that before were in the preclinical, right now they are in the clinical domain. We have more than ever phase 3 molecules that are very close to the FDA approval. Some of them are alpha particles, some of them are beta particle emitting, and it alone shows what a thriving field we are dealing with. But is it truly a last mile that we need to tackle? Is it fair if we say the only thing that is between us and success in this domain is just like the last mile and we should go there? I don't think so. I feel readiness is extremely important. We need to be ready to not only use this opportunity, this very important momentum that we have, but also build on top of that. As Arman mentioned, we realized that access to these radiopharmaceuticals at the clinic, when the patients are living in the mainstream, is a big challenge. There is a difference between availability and accessibility. And to bridge the gap between the availability and accessibility, we need a network of the reliable clinical operations across the US that can bring high quality practice of medicine to the patients that they need. And access to these radiopharmaceuticals in both clinical and clinical trials are very important. But we realize this matter is not as easy as we think. In order to elaborate on that, and I know most of you are very familiar, but because we have also online audiences, I need to very quickly say the radiopharmaceuticals are the molecules that have a radioactive isotope in addition to a targeting molecule in some sort of shape. And there are many classes of these targeting molecules, from very large molecules to the smaller molecules, and they can be combined with various radionuclides. And one fundamental issue that we are dealing at this moment is the concept of radiopharmaceutical itself. Is it chemistry or is it physics? Is it something that emerged as a pharmaceuticals or as a radiation source? And maybe none of these two. And then I tell you why this is not just like a semantic splitting of the concept. It's not just pharmacotherapy because the effect is through radiation biology, and it's not just radiation therapy because the source is not determined by us, it's by the biological system. There is a very important spatial domain heterogeneity, very important temporal domain problem, and the biological domain is also extremely important, importance of the immune system and bystander effect and organ at risk primacy. So again, that created a lot of confusion because people think that's radiation therapy. So let's just like do whatever we do in the radiation therapy here, while we don't pay attention that the reason that we have word pharmaceutical there is because the location of these unsealed radioactive material is not determined by us or the therapy physicists. It is completely a dynamic way that localize the lesion. I'm not going to elaborate on that multiple times. We talked about this concept of radiopharmaceutical therapy as a complex system, multi-scale, which needs a multidisciplinary approach. And I'm going to just like today talk about some very interesting conflict between the impact of chemistry and physics in medicine. As you all know, many of the chemical or medicinal products that are used in medicine coming from the textile industry and the coloring of the textile. So, and chemistry for a long period of time was perceived and is perceived as the modern medicine, in contradistinction with the traditional medicine, which was physics. So I didn't create that myself. Last night, out of surprise, I found this by searching the literature, that somebody just like concluded that when physics start interacting in medicine, that was a traditional medicine. And then in a new modern medicine is a time that we have chemistry coming and doing that. And of course, I mean, whoever this another group will receive something back. So the physicists say that I feel bad for you for saying that. And the chemist said, I don't think about you at all. And this goes on and on. To just like put it in perspective and see the funny dynamic between these two groups. Look at the Sir Richard Feynman, which concluded chemistry is physics without thought. And that's not there. Ernest Rutherford was just like concluding that physics is the only real science. The rest are just stamp collection. And the funny thing is that he received a Nobel Prize in chemistry himself. So again, the perception between these two groups are not easily reconcilable. They have some deep problem with each other, and I cannot ignore. And these two worlds need a lot of work to work with each other. But again, remember, science is collaboration, which means all of us get to fail together, publish together, and blame the grad students when it goes wrong. So we are all united in this. However, we all know that there is something different about radiopharmaceuticals. The new guidance on a dosage optimization, very recently published by FDA, August 2025, shows that even regulatory agencies don't know exactly what to do with this process. They know it is not pharmacotherapy. They know it is not radiation therapy. They don't know exactly what it is, and that's painful. So we have opportunity till 20th of October to respond to this opinion. And that's very important because beside the regulatory bodies, finding the molecules that have a higher chance of success is a very important factor that decreases the cost of the new drug development. And that is very important if you look at the chance of the early target detection to the FDA approval or regulatory approval, market authorization of 1/10,000 molecules. So again, finding the losers sooner might be very financially important for the people who are even developing the treatments. So that was the reason we realized just having united Theranostic is not good enough. It's an extremely important factor, but not good enough. We realized that in order to make the access a real access, we should focus on meaningfulness of that access. Otherwise, it creates pseudo access. You may just like see the hunger in a society, and instead of just like bringing good food, you may just like bring junk food. And that just like conceal the lack of access by pseudo access. So it's very important to look at meaningful access, and not only meaningful access, because if you have also meaningful access, but don't think about the ecosystem that this meaningful access live in, it's not sustainable. So sustainable meaningful access was the intention behind the creation of the nonprofit organization with the name of Institute of Nuclear Medicine, which is laser focusing on the nuclear oncology and Theranostic. And it has 7 grand challenges. How to optimize the radiopharmaceutical therapy. And I'm lucky by having great friends like Carlos and Arman and many of you here, that during the past 3-4 years, we pushed this concept of computational nuclear oncology and Theranostic digital twin ahead, or the concept of dosimetry in a simpler way. But after optimization, you need to find the lack of access in the society. When you find that, then you need to have a method to intervene in a way that do not cause more harm. You may, with a good intention, you may just like come to a location in a geography, do something, and it come back as a surprise to you that not good intention is necessary but not sufficient. So implementation and dissemination science is important. But that's not good enough, because even if you do that, sustainability requires training of the next generation. We need to focus on that. Otherwise, it will be just like a very bright moment for a short period of time, and it goes down to the darkness. And no group alone can do all these things together. You need to share, you need to collaborate, and that was one of the reasons of the Institute of Nuclear Medicine, to gather, for example, information, clinical data, be a neutral third party that enhance and augment the collaboration. Then we realized that all of these good things, if we are not focusing on advocacy, like the interaction that we had with the FDA, like the sending the comment to this guidance paper, it's not going to be implemented to the way that we want, with the payers, with the regulatory agencies. And at the end, very important concept of trustworthiness. Many people think trust and trustworthiness is a byproduct of the good intention and good people sitting around the table, and then suddenly very good trustworthy situation happen. It's not the case. Trust and trustworthiness needs to be designed, and that's the active construct of an operation. And we are committed to that. And these 7 challenges were the beginning of the INM journey. So going to the radiopharmaceutical therapy optimization, again, this is one of the slides that multiple times I taught, and I'm not going to stay there. I'm just like showing that to move on. We want to optimize the patient selection. We want to optimize the delivery of the radiopharmaceutical. What's the dosage? What is the interval of that injected activity? We want to minimize the biological resistance and maximize the immune response to the therapy. Of course, we want to use a technology like AI to measure the microscale dose to the organ. Of course, we understand there is a big difference between microscale image-based dose measurement and the microscale effect, because the range of these particles have a range that is very important in biology. The cells are 20 micron, 30 micron, 10 micron. So there is a big difference between 40 micrometer range and, let's say, 400 or 4,000 micrometer range. This is important to understand. Even in one category of the radionuclide, the range is significantly different, and that might be one of the factors that shows the different impact of yttrium with the same target of dotatate versus the lutetium for that. Of course, we know that the biology lives in the scale that is in the micrometer scale, and it's not only for the kidney, as we saw in the previous slide, it is also for the bone and bone marrow. The trabeculae and the space that we are living in lives in just like 20, 200, 2000 micrometer. And that makes a big difference. Heterogeneity matters because the biology lives in that scale, in the spatial domain. Of course, we know that the temporal aspect of the radiopharmaceuticals are also extremely different than external beam radiation. We are dealing with orders of magnitude, 4 or 5 order of magnitude different rates of the radiation. And that is not only the rate, but also the duration of exposure, which is very different between radiopharmaceutical therapy and the external radiation. So a few years ago, as a thought model with the help of friends. We put this concept of Theranostic digital twin in place as a roadmap to bring these concepts into one mental framework. And we realized that cancer is complex, it's multi-scale. We need to use the prior aware knowledge and data. We need to integrate multiple systems to each other. We need to have combining the macrodosimetry with the micro scale radiobiology and computational biology and computational oncology. We knew that we have to consider tumor heterogeneity as a very important factor, and also multimodality approach capture of that. And the precision radiopharmaceutical as a complex data science at core within knowledge domain of the nuclear medicine that require a fundamental shift in the way that we are thinking. The thing that I'm going to talk today mostly is about the approach. The approach is that you can approach the same phenomenon from two different perspectives. One is just like bottom to top. Just like see what is the microanatomy, how is the structure of the kidney, what are the receptor densities, what is the receptor internalization. How you can really learn from the microscale dosimetry, and then with that knowledge reach to the level that how much you can just like change the administered activity, for example. The other way to think about that is top to bottom. Just look at the clinical cases. By mere fact that we are injecting the same amount of radiation, injected activity, same amount of radiation activity to the patient, that results in various and variable dose to the organs. See how that impact the function, how we can learn from that top to bottom approach and looking at the real world clinical data to conclude and extract the unknowns. That's a very important matter. I'm going to allude to that when I'm going to the clinical computational nuclear oncology. But before going there, and I was advised yesterday that I should not just like too much talk about the clinical aspect and more about the clinical computational. But I want to say that at INM, we have task groups that are, from medical standpoint, laser focusing on the organ at risk management when the damage happens, or how we can quantify the damage. These are the task forces that we have already. And in September 26 at 6 p.m., we have a session on nuclear onconephrology. Onconephrology is a field in nephrology that focus on impact of the chemotherapy or other oncology treatment on the health of the kidney. So how we can build on top of that knowledge and from the clinical standpoint, focus on impact of radiopharmaceutical on the function of the kidney. For example, I see many of the clinical trials and many of the even non-nuclear medicine practices that are following the function of the kidney merely by creatinine or the GFR, while forgetting creatinine without the albumin creatinine ratio of the urine is a very, very limited, if not useless, biomarker for the health of the kidney. So how we cannot forget ACR is an important fact. That's not just like there. We have also other clinical trials going on that how we can use various remedies like bromiplastin as an enhancer of the thrombopoietin thrombocyte generation for treatment of the radiopharmaceutical induced cytopenia. So why we should ignore that? I mean, if there is a side effect, if for a specific reason one of the radiopharmaceuticals may cause a damage to the megakaryocytes, we have these thrombopoietin receptor agonists that can activate, and there are very good result on that. But I put that aside and go to the clinical computational nuclear oncology, which I say the second challenge after the purely computational. And that is the point that I want to emphasize. Again, the intention of that is using the real world data for addressing some of the complexities that in a micro scale might be very difficult to address. And that's not just like a phenomenon that is for this multi-scale complex system. If you look at, let's say, thermodynamic and the gas physics, if you want to measure the velocity of each molecules alone, it's a very difficult challenge. But when you look at the statistical mechanics, if you put a thermometer inside a chamber, by just measuring the temperature of that complex system, you can have a very good idea about the average velocity of the molecules in that chamber. Sometimes you have an emergence and manifestation of a complex system that, although you don't know the microscale aspect of that, you can observe and manage in a macro scale at a different level of certainty. This is not even new in nuclear medicine. For a long period of time, we practice this concept in a different way. So, of course, when we are looking at the micro scale dosimetry, we know that what a difficult job we have, how to go from the understanding of a nephron and radiobiology of that. And these are very, very important. But how we can define biological effective dose of radiopharmaceutical therapy, which is not at all relates to the biological effective dose of the external beam radiation. There is no alpha, there is no beta, there is no, those assumptions that nobody knows exactly comes from there. And this is the thing that in nuclear medicine has a history. When people wanted to do still at NIH, we were using dosimetry for radioiodine therapy for thyroid cancer, particularly patients. We had a program for patients from Ukraine after the Chernobyl incidents with very retractive and retractable thyroid cancer to come to the program. And we realized that without a very well designed dosimetry-based radiopharmaceutical planning, radioiodine planning, we may create a lot of resistance. But how it happened was just like about the retention of the radioiodine in the lung. They didn't know exactly what is the dose, but they knew that if certain amount of activity retained in the lung in 48 hours or 72 hours, that has a dire impact on the lung function. Radiopharmaceutical therapy biological effective dose is comparison of the effect of a new type of radiopharmaceutical on the function of the organ compared to some reference standard. You can just like consider that as a theoretical framework. And that shows in order to have that biological effective dose, it depends on A, what is radiopharmaceutical that we are talking about, B, which organ that we are talking about, and maybe even C, what is the reference that we are talking about, because we want to manage that. So if we look at this quest, we want to collect enough information, looking at the patient, different baseline of the organ functions there, certain assault or exposure happen, and then you have a change in the function of the organ, and you want to reconstruct the reality out of those information. But the amount of information is astonishing. You have multiple injections, you have multiple PET CTs, multiple SPECT CTs. The result of those image processing resulting in the dosimetry is there. Lab results related to the function of the organ at risk is also coming. and the drug information, the device information, the process information. Very soon, if we don't have a model that capture these things in a very organized manner, we have a sea of data without seeing the important matter that is in front of our eyes. And that is my point for call to arm. We need to design our clinical practice as a large observational clinical research to capture the capturable to the level that we can. If we cannot do 2 times dosimetry, SPECT, have 1 time. If we cannot have multiple PET, have 1, but at least measuring the renal function in the right way and following that in a way that you can retrieve that later in order to conclude and drive the clinical inspect on that. And that's not the first time that we have. Real world data is in general very, very important. And many of the regulatory agencies even mandate that for some of the treatments that are not radiopharmaceuticals. And that's difficult because you have various data models from different sources, diagnostic imaging, clinical phenotyping, treatment, etc., and the data from the clinical trial that have a different data model. And how you are going to combine these things with each other. So I want to move to this universal ontology for radiopharmaceutical therapy for standardization of data. And that is my main message today, that we need to move, because although from bottom to top approach is extremely important, these mechanistic models from multi-scale modeling is extremely important, and the construct of clinical nuclear oncology, if we ignore the fact that we need the top to bottom, or just like from patient to outcome, we miss a big opportunity. The complexity of the system is very higher than what we can imagine even that stage. So what are the ontologies that are in medicine that want to kind of harmonize the way that we communicate? So from early 20th century, that international classification of disease by WHO started to classify the diseases, to mid 20th century that for payments, there were just like nomenclature about terminology of what we do with the name of CPT, which is current procedural terminology for payments, to the concept of NDC, National Drug Code, that uniquely identify not only the medicinal product, but how it's get packaged and how it is manufactured. 2 very important events in medicine, which was creation of the SNOMED CT, systematized nomenclature of medicine, and evolution by combination of multiple ontologies and adding very important knowledge modeling concepts, which allowed the fact of multi-hierarchical construct and the semantic tags. It led to creation of the logical observation on identifier, name, and code, LONC code, that right now without LONC code, you cannot just like communicate with your lab. If you want to just like have a CBC, you don't know what is exactly that code. You cannot just like have that. As a point, don't we do in dosimetry similar activity? Why we should not have a similar observation identifier, name, and codes? And we will talk about that. So, of course, the lexicon of the for radiology, the RADLEX was there, and looking at the need for these, NucLex was a concept that was initiated about 3 years and a half ago, and the aim of that is creating an ontology for radiopharmaceutical therapy to combine different aspects of need in Theranostic. And I want to just like emphasize on three aspects. NucLex is, in addition to the ontology, is a data model and process model. And I tell you what are the difference between these two. So an ontology is a construct that you connect different concepts with each other. Each concept has a definition, which we call that describer, and it has a relation. And also, you can just like find that in the web of semantic relation with the other. The best example that we found for building the NucLex ontology aspect is on the SNOMED CT. SNOMED CT has about 19 hierarchies, and we have this opportunity to build whatever is not there on top of that. But that's not the case only for ontology. Process is also extremely important. One of the aspects that process is getting modeled is in clinical trials. In clinical trial, when you are submitting the data to the FDA, you must have a very, very rigorous way of the data architecture. And in addition to the just like submission, you have even the planning of that clinical data flow can be captured in a very rigorous manner. If you have a dosimetry practice, how it goes, how you just like find the first image, what is your process of doing registration, extraction, and how you can make sure that through that process you genuinely incorporate a quality management system that, like any GXP, good X process, like good manufacturing process or good laboratory process, this time is a GDP, good dosimetry process, and bring those concepts to the forefront. And the third one, in addition to ontology, second, the process mapping or data model, and the third one, the clinical data model. How we can just like see to connect the database that we have with the HL7 or FHIR database that our EHRs are living in. Theranostic is not just like imaging. If we only focus on the imaging informatics and forgetting the importance of the clinical informatics aspect of that, we are missing a big portion of that. And how we can combine these through the concepts like the OMOP. So again, this not only create a platform for us to gather the information for breast cancer for the radiopharmaceutical. In the past, it had been used for breast cancer. It had been used for the for many of the other chronic disorders, and we hope that this aspect bring very important role for building the top to bottom aspect of that. And I'm just like ending that with one simple example of how process mapping in a measurement is a very important factor in the clinical practice. If you don't know exactly what was the geometry of your dose calibrator, and you don't capture that, how you can rely on the measurement that coming out of that? If you don't know about the radionuclide purity or radiochemical purity or specific activity, how you can incorporate that in the in your conclusion? How we can move from just like a one task metrology to the total life cycle of the data that include all the steps that are necessary for that one number and that measurement. So emphasizing on the imaging informatics and clinical informatics under the concept of NucLex is one of the very important aspects that at this stage we believe is needed for addressing this unmet need and moving toward meaningful access and sustainable meaningful access. So I'm very grateful for this opportunity to talk to you all about these upcoming challenges, and I'm happy to answer the questions. And again, at the end, I want to just like emphasize all the things that we have in our armamentarium are just simple tools that we use for caring of the patients who need that. And it is our obligation to use whatever we can to make them not to suffer or suffer less. Thank you.